Good evening to our chief guest of the occasion, Professor Nalma, Vice Chancellor, Jamia Millia Islamia. Our guest of honor, Dr. Rafi Ahmed, Director, Emory Vaccine Center, and our special guest, Professor C.C. Khalid, Dean of Agri Sciences, Jamia. In view of the COVID-19 pandemic, it is ostensible to have virtual format for celebration. The advantage of this format is that it makes the program accessible to many more people in their own comfort and ways. We are also going to have separate session for press and media with Dr. Ahmed. The links are being shared. To inaugurate the Centenary Lecture, it is my great honor and pleasure to invite our Honorable Vice Chancellor Ma'am to enlighten us with her views. As an advocate for gender equity, Dr. Akhtar is the first woman to hold the post of Vice Chancellor of Jamia Millia Islamia. Prior to this, she was the professor and head Department of Academic Building in Education, National Institute of Educational Planning and Administration. She has led several international consultancy programs, authored 2 books, and contributed numerous book chapters and research papers. She has been very actively involved and mentoring research and academic activities. Jamia has achieved phenomenal accolades during her tenure. Now, we are among top 10 universities in the country as per NIRF ranking and in the top among central universities. May I now take the liberty to request our Vice Chancellor Ma'am to say a few words and commence our event. Dear Ma'am, please.
Thank you. Thank you for giving me this opportunity. Can you hear me? Am I audible?
Yes, ma'am. Yes, ma'am.
All right. Okay. So, Dr. Rafi Ahmed, Director, Emory Vaccine Center, Atlanta, USA. Professor Simi Farad Basir, Dean, Faculty of Natural Sciences, Jamia Millia Islamia. Professor Zahid Ashraf, Department of Biotechnology, Jamia Millia Islamia, and other distinguished guests, teachers, dear ladies and gentlemen. A very good evening. And Dr. Rafi Ahmed, is it a good morning there? I'm sure it is morning time in your place. Good morning to Dr. Rafi Ahmed and all the participants across the globe. I welcome you all to the centenary celebrations of Jamia Millia Islamia and to the centenary lecture on a very relevant topic on immune memory, remembering your infections and vaccines. The current coronavirus pandemic has drastically transformed the entire world. It has changed the way we live, we socialize, and travel around. The pandemic has literally paused our lives, but this pause offers us a rare opportunity where we can sit back and ponder on the things that went wrong. Why we have landed in such a situation. Now we have time to sit and think. It is also time to reevaluate our ideas and restart investing in healthcare systems and scientific research and development for a better future. At Jamia, we all know that it is the 100th year of establishment of this great institution. Therefore, to commemorate its magnificent and extraordinary history that dates back to 1920, we have organized the centenary celebrations, and that Dr. Rafi Ahmed Saab today is also a part of this celebration by coming to us and by his very relevant and eminent lecture. The ideology and principles of the founders of Jamia Millia Islamia made the university an epitome of academics and research excellence. It has 39 departments, 27 centers, and 7 schools. And Jamia Millia has become a country's top leading central university, which lays due emphasis on expressions, learning, innovation, and of course on character building. The distribution of exceptional faculties and their findings and their contributions have increased the visibility of their respective departments, because each faculty has a number of departments, each one doing in their work and research in different areas, and these departments are very much visible, and the university is also visible because of the teachers who are doing research in the whole world. Their continuous efforts have brought our university into a league of premier institutions in the country as well as in the world. The Department of Biotechnology, Jamia Millia Islamia, which was established in the year 2008 with the broad vision of carrying out high quality multidisciplinary teaching and research in the area of life sciences. The goal is for overall development and betterment of the health, longevity, agriculture, and biotech and pharma industries. Innovation and technological advancements have been essential and very useful for fundamental and biomedical research and biotechnological industries in general. Our Department of Biotechnology, which has organized this program, at present has more than 20 research projects for the amount of more than 10 crores, expanding in the area of genomes, proteomes, and both fundamental and translational research on communicable and non-communicable diseases. Recently, the department has also been awarded the financial support from the Department of Science and Technology, Government of India under FIST Level 2 for the year 2018. It's a great honor today and pleasure to have Dr. Rafi Ahmed with us to share his thoughts on his remarkable scientific journey, his vision for Emory Vaccine Center, and its work in India. His centenary lecture today will cover immune memory, remembering your infections and vaccines, and his presence uh, will be good for our teachers as well as for our students, because seeing a person who is actually involved in such a level of research makes you inspire you, and you remember them for a long time and try to go on the same lines. So I'm grateful that Dr. Rafi Ahmed has found some time to be with us, and it's a great honor for this university. Dr. Ahmed, as I know, is an incredible virologist as well as immunologist, and is sought after worldwide as a consultant and a speaker. And the ingenious and enterprising nature of Dr. Ahmed is not just limited to the immunological discoveries in the lab, but also in the creation of an internationally regarded vaccine center where the most comprehensive vaccine discovery studies are conducted. The Emory Vaccine Center is now recognized for the development of vaccines for HIV/AIDS, hepatitis, malaria, tuberculosis, and influenza. But even after all these awards and accolades, I have been told that he is still the happiest among his students and colleagues, sharing his knowledge and wisdom with all, as he knows that the humanity at large will reap the benefit out of it. While with the same thoughts, I would like to welcome Dr. Ahmad to Jamia, to India, and I take this opportunity to request Dr. Ahmad to continue his association with Jamia teachers, Jamia researchers, and students. His mentorship and guidance will help the younger faculty, students, and university to achieve greater excellence and excellence in research and development for the university, for the country, and for humanity. In the end, I must say that we are all very pleased to have him here. And I personally would like to offer him a position of chair in the upcoming JMI Vaccine Institute. We now request Dr. Rafi Ahmed for his address and to make this centenary celebration of Jamia Millia even more remarkable and more memorable. Thank you very much, Dr. Rafi Ahmed. Thank you very much.
To achieve our ambitions and aspirations. You are a continuous source of inspiration. Now, with the permission of Vice Chancellor Ma'am, one of our student representative, Ms. Azma Nasir of the Department of Biotechnology, Jamia, will present a token of appreciation in the form of portrait to Dr. Ahmed for accepting our invitation. This portrait was made possible with exceptional support from our renowned faculty of fine art at Jamia Millia Islamia. Ms. Ahmed, please.
Very good. I thought this is very good. Okay.
Yeah, okay. Okay, I want to thank the Honorable Vice Chancellor for this very kind introduction. Can you all hear me?
Yes.
Yeah. Again, as I was saying, I'd like to really thank the Honorable Vice Chancellor for very kind remarks. And I would like to thank Jamia Millia Islamia really for this honor of being part of their centenary's celebrations. It's an institution that all of us have greatly admired over the years for their great contributions to education and to India. What I'm going to do today is divide my talk into two parts. I will first start with a brief overview of the Emory Vaccine Center. And then I will discuss basically my work. And I'll do this at a high level. And I've just titled this part a virologist's journey into immunology. I will then end by an overview of what is currently happening in COVID-19 vaccines, where vaccines are in terms of development and in terms of being used. So our vaccine center was established in 1995 and is now one of the largest academic-based vaccine centers. Our mission is to improve human health by conducting both fundamental and clinical research leading to development of effective vaccines, and not only against infectious diseases, but also against chronic diseases like cancer. The key part of our, the key success, I would say, of our vaccine center has rested on how we initially set it up. And we set it up really as from a soups to nuts operation. And when this was set up in 1995, it was very rare to have a vaccine center type organization at an academic university. Certainly academic institutions are not pharmaceutical companies which have manufacturing capabilities and so on. But we felt that having an academic-based vaccine center can greatly contribute to the vaccine sciences and to the development of effective therapies and vaccines. And we also felt that when you set up something like this at an academic institution, it should really be very interdepartmental. And we didn't want to just create another department of vaccine science. It really had to bring people from different disciplines together and working in a very cohesive way. So we set it up as really always the fundamentals of any kind of scientific activity really is the basic science. So we recruited faculty which had expertise in vaccine discovery, immunology, pathogenesis. We firmly believe that if you want to make a vaccine against a particular pathogen, you need to understand the pathogen itself. You need to have expertise in it, in its structure, how it replicates, but also in how it causes disease. So that really this pyramid rests on fundamental and basic science. And then we have selected programs so that there's focus on specific pathogens, and we kind of set up a center which kind of focused on some of the important diseases that we currently have. And then we felt that it's really essential to have a clinical arm of the vaccine center where it's possible to do clinical trials, do human studies. Another focus of our team was, even though we do want to study basic science and fundamentals and mechanistic studies, we also want to do human immunology and really study the human immune system. And this required that we had a facility that would allow us to do human studies. And then we were very fortunate in recruiting Dr. Walter Ornstein, one of the world's leading vaccinologists and expert in vaccine policy. And so we felt that even going beyond this, we should have some expertise and input on vaccine policy. And we've been very fortunate over the past 20 plus years in having truly outstanding faculty. And we have faculty from many different departments, many different divisions. There are clinicians on it, there are basic scientists, and people from public health, and people who have expertise in vaccine policy, as I mentioned. And this is a wonderful group of people that have worked together. So the secret to our success actually is this slide. We work as a team. Scientists are competitive. And we respect their independence. We want them to develop their independent programs. But for the questions we were asking, it's difficult for one individual or one faculty member to achieve these. So basically, team science really became our motto. And then do fundamental discoveries here and have translational impact of these findings. Currently, our programs are focused in these areas. There's a very large program in trying to develop a universal influenza vaccine. As you all know, influenza virus mutates, comes in many different shapes and sizes. That's why we keep changing our vaccine every year. The goal here is that we develop a vaccine that will protect us against many different strains of influenza, and really one wouldn't even need this annual vaccination. This is a very ambitious goal. It will not be easy, but this is a major program currently at the Vaccine Center in collaboration with the other institutes which have joined the program to develop a universal influenza vaccine. We've always had a great interest in emerging infections of global importance. Emory was a place where 4 of the Ebola-infected patients, when we had the West African outbreak came, and we were able to do studies on human immune responses in Ebola patients. This was a unique opportunity that we provided to us. And currently, of course, we are all focused on COVID-19, and there's several people working at the Vaccine Center on COVID-19 vaccines in the area of studying the human immune response, in terms of making human monoclonal antibodies, in terms of being very active participants in the ongoing vaccine clinical trials, both in the phase 1, phase 2, and the phase 3 clinical trials, in addition to doing research on optimizing vaccines. Perhaps the largest program of the center is really HIV vaccines and HIV cure. And here the goal is not only to have the preventative vaccine, which probably is one of the toughest challenges that we face in the area of vaccinology. This is probably the hardest virus to make a vaccine against because of how it mutates, but also the way its structure is, and it's a very difficult target that we are working with many other institutions to do that. In addition, we are working on a functional cure so that people can get off the antiretroviral drugs and be able, the immune system to control the Chinese infection. A major program that we are currently working on in collaboration with the Millia Cancer Institute at the university is cancer vaccines and immunotherapy. Another program headed by microbiologists is the concern with antibiotic-resistant microbes and whether actually one could have vaccines against these, and enhancing global vaccine use and advocacy. Because this is an important activity, because if you have a vaccine and you don't take the vaccine, really it's of no value. So it's important that not only the important vaccines be developed, but then they are then used and distributed and given to the public. And then we also have a very active international program. And we have collaborations in Africa, South America, Australia, Asia, and most importantly, in India. And I'll just spend a few minutes telling you about our program here. We were very fortunate to enter into a wonderful partnership with ICGB, International Center for Genetic Engineering and Biotechnology. I'm sure you're all very familiar with this wonderful institute. And this partnership was initiated several years back and was really supported not only by Emory University, but by the Government of India, the Ministry of Science. You can see here in the opening, Mr. Rafi Ahmed actually doing the ceremony of lighting the lamp. We had great support of the Department of Biotechnology and the ICGB leadership in setting this up. The focus of this center that we have located at ICGB really again is human immunology and addressing infectious diseases that are a large burden in India and in that part of the world. And I'll just tell you a few words about the major program that we have currently here, which is looking at dengue infection. So dengue virus infection in India is a major problem. It has been a major problem for the last 25 to 30 years. And unfortunately, the largest number of infections worldwide on an annual basis are in India. There's about 100 million cases worldwide, and India has the largest number, and the estimate is that between 7 to even as high as 20 million or so cases could be in India. And there's active efforts going on at many different places to try to develop effective dengue vaccines. Currently, many clinical trials are also going on in India. Indigenous dengue vaccines are being made at ICGB, which initiated at ICGB by Rafi Ahmed. Those are also going into preclinical and hopefully soon into phase 1 clinical trials. Our center has really been focused on understanding human immune responses to dengue virus infection. And we are very fortunate in getting a large NIH grant and support from the DBT to really start addressing this issue in great detail. What happens during the primary dengue infection? What happens during the secondary dengue infection? What are the potential bases for perhaps enhanced disease during secondary infections? And for this, we have set up a very nice collaboration with many clinicians. And so basically, we have our lab at ICGB. We are interacting with the pediatric department and clinicians at Ames Hospital. We also get samples from the Christian Medical College. And we've also been collaborating with the National Institute of Virology in Pune and with scientists at USSDI. And several papers have been published on characterizing the immune responses. And in particular, we are interested in understanding the differences between primary dengue and secondary dengue infection. This is going to be highly relevant for the development of the vaccine in India and during these clinical trials. And this is just a photograph of the group. Every year we get together in Delhi and we have a meeting of people who are part of our dengue consortium. And this is one of those events where you see not only people from our lab, but also some of the people from who are our collaborators. I just want to highlight here two individuals, Anmol Chandali, who's kind of heading our lab at ICGP, and Murali Krishna, who also works very closely with Anmol in running our program. So let me now switch to telling you at a high level really some of the work that we've been fortunate to do in our lab over the past 7 years. I did my initial training in my PhD in virology, so I really was trained as a virologist. And so what I'll highlight in the next 15, 20 minutes is really a virologist's journey into immunology. And even though my training was as a virologist, I really got very fascinated and interested in the question of immunological memory. And when I started my lab at UCLA in 1984, this was my first independent position, I decided to really focus on this issue. How do we remember the previous infections that we got? How do we remain protected from infection? What is the cellular basis for this protective immunity? And I was especially inspired, I suggest that's the right word, or appreciated the importance of this based on a wonderful experiment of nature that happened over 200 years ago. And in the next few slides, I'll describe to you this amazing experiment of nature that happened. And this experiment of nature occurred on a small island called the Faroe Islands. This, as you can see from how it is very isolated. You've got Iceland here, and you have got the Europe mainland over here. This is right in the middle of nowhere. At this time, the Faroe Islands was actually under the government of Denmark. And in the next slide, I'll describe to you this measles infection that happened and how this experiment of nature really allowed us to gain very interesting insights about immunological memory. So here's the experiment of nature that happened. In 1781, there was a significant measles epidemic on the Faroe Islands. And then what had happened in this ensuing years, 1782 to 1845, is that, I'll go back to the slide again, that the Danish government, I don't know the political reasons for it, but the Danish government had imposed restrictions on travel between mainland Europe and the Faroe Islands. And so in the ensuing this period, there were really no reported cases of measles because a lot of virus was going back and forth in the past between the small Faroe Islands and the rest of Europe with no travel and completely isolated. There was really no more measles on the island. And then these restrictions were kind of decreased, basically, then travel started between mainland Europe and the Faroe Islands. And in 1846, there was a major epidemic. And this epidemic of measles was so severe that up to 95% of the population got measles, 70 to 95%, almost everyone there got measles. Measles actually is a terrible disease, also has a high mortality rate, and has high risk, 15 to 20%. So when this epidemic happened in 1846, there was a Danish physician, actually he was at that time a medical student, Peter Ludwig Panem. And Panem went to the Faroe Islands in 1846 to investigate this measles epidemic. And then he published a remarkable epidemiological study in 1847. And this actually was part of his thesis work for his medical school graduation. And he made many important observations. And this actually was, if I would highly recommend people who like to read classic papers to kind of go back and look at this. You can find this has been reprinted in this medical classics, and you can find it there. But really, the original paper was published in 1847 after he did it. And he made two amazing observations. He found that of the many aged people still living on the Faroes who had had measles in 1781, not one was attacked the second time. So these were all old people who had had the measles in this, they had had measles in 1781. And in 1846, 65 years later, they were all protected. Not a single one of them got measles. He also had an amazing control group because there were some old people who had not had measles in 1781, and all of them got infected. So what this proved remarkably is that protective immunity for 65 years could be there in the absence of re-exposure. And I really want to highlight this part. We've known since the time of the Greek, 3,000 years back, that somebody who got the plague usually did not get plague again. But of course, you were always being exposed to whatever was causing these diseases. But here was an experiment where there was no exposure to measles virus for 65 years, because there were no cases of measles, this place was totally isolated, and these people were protected. So what this said is that our body has intrinsic mechanisms that can confer protective immunity, at least in the case of measles, almost for 65 years or longer. And this is something that our immune system is doing without any re-exposure to the pathogen. And this is the story that really fascinated me. And when I set up my lab in the 1980s at UCLA, really try and understand what is the mechanistic or the cellular basis for getting long-term protective immunity. So of course, one starts with basic animal models to study fundamentals and mechanisms, and then you extrapolate those studies into humans. So what are the mechanisms of protective immunity? There are two major mechanisms. So the most important mechanism of protective immunity is having pre-existing antibody. That is antibody that's already present in your blood, in your circulation, and hopefully also at the mucosal sites. So when the virus that comes in, immediately virus is neutralized and the infection is either minimized greatly or even completely reduced. So pre-existing antibody is really our most critical parameter for protection against infectious disease. And then this important second line of defense are your CD8 T cells and also CD4 T cells, in particular the cytotoxic CD8 T cells that will eliminate any infected cells that have happened, because very often utilization is not perfect. So some cells do get infected, but if you have a very fast and rapid response from your killer cells, you can eliminate those cells. So let me first address this issue of how can you get antibody for years, persisting for years, without even re-exposure, as was shown in this wonderful epidemiological study of pain in the Faroe Islands. And so when I entered this area of investigation, the dogma was, and this was what all the textbooks said, is that small bits and pieces of antigen and viruses persist in the individual, and very hard to prove that this does not happen. Proving the absence of something is very difficult. And that this is what kind of keeps perking up the immune system. So you're getting some low-grade stimulation. And one cannot rule this out, obviously, after a live infection. Small antigen might persist in small numbers, and that keeps stimulating the immune system. And this becomes very important because the half-life of antibody in the serum or in the body is only a few weeks. And within 2 to 3 weeks, antibody will start decaying. So the only way one could maintain long-term antibody responses are either by some antigen stimulating, or if there was a cell that could produce this antibody and be long-lived. So the cells that produce antibody in our body are plasma cells. This is the question: how can antibody responses in humans how do you get this long-term antibody? And is there a cell that can produce and live for very long? Antibodies produced by plasma cells. Plasma cells are a very interesting cell type. They have differentiated and are continuously producing antibody. The only function in life of plasma cells is to make antibody. They are a factory that's making antibody. These cells don't divide, and it was assumed at that time that they were also very short-lived, that they die within a few weeks. So the dogma at that time was that the plasma cells that make antibody are short-lived and die within a few weeks, and the antibody, the molecule itself, of course, had a short half-life. So how can you get this thing? So work that was done in my lab by a graduate student, Mark Slifka, actually showed that there are indeed, not every plasma cell is long-lived, but there is a subset of plasma cells that can live for very extended periods. Mark's studies were done at that time in mice. These studies that we did, and we showed that actually in the absence of any replenishment from these cells, these plasma cells could live for the life of the mouse. These studies have been extended by us and others that in humans also we get very long-lived plasma cells. Also, Andreas Radbruch in Germany made similar observations. So this discovery and identification of the long-lived plasma cells really provided an explanation for long-term protective immunity. It also becomes now a goal for vaccines. That is, the best vaccines should be the ones that are able to generate these long-lived plasma cells. And there are mechanisms of how these can be generated that we and others are studying. So this discovery not only provided a cellular basis for long-term antibody production, but has provided guidelines in terms of how our vaccines can be optimized for giving long-term durability. So typically, this is what the response looks like. Antibody goes up during the acute phase of infection because there are plasma cells making antibody, but many of these are short-lived. They will die. But then you reach this stage where you have long-lived plasma cells, and the location of these long-lived plasma cells is very interesting. They are mostly found in the bone marrow because this is where they get the nurturing signals for their long-term survival. And this you can get persistence of these long-lived plasma cells for many, many years. So what about the T-cell immunity? Well, how does the T-cell contribute to protective immunity? We have two major subtypes of T-cells, the CD4 T-cells that provide help for antibody responses, and they also have their own antiviral effects as effective functions. And then we have the CD8 T-cells, which are your killer cells. These are machines that are really trained as killers. The CD8 T cells are the most efficient cell type for eliminating virally infected cells and also for eliminating cancer cells. These cells can go to the site wherever the infection is or the tumor is and kill those cells. So you've got the early defense against infection by pre-existing antibody, and then you've got these killer cells that can come and kill the virally infected cells. So this is how the curve typically looks like for a T-cell response upon infection vaccination, especially if you have the right type of vaccine. You get an increase in the number of the virus-specific T-cells. Many of these will die, but then again, you get these long-lived memory cells that can persist for decades. Work that was done by another graduate student in my lab many years back, Lisa Lau, showed that this persistence of the memory T cells, again, does not depend on antigen. These have intrinsically long-lived cells that can also undergo some slow self-renewal capacity. So the process of longevity and some slow self-renewal keeps these cells living for decades. Work that has also been done by Lauren Krishna, kind of identified this pathway of expansion, contraction, and long-term maintenance. So what are the properties of memory cells? Why are memory T cells different from just a naive T cell? So one of the most important characteristics, as I mentioned to you, is their long-term survival in vivo. these memory cells can persist essentially for decades, if not longer in humans. As I mentioned, they're capable of slow homeostatic proliferation to undergo some self-renewal. And then this is the most important property of these cells in terms of why they're very effective in controlling infections. Because these memory cells, unlike a naive cell that takes time to develop effector function and become a killer, these cells can become effectors very quickly. Basically, they're able to kill cells, infected cells, literally within minutes, within hours of exposure to antigen. So their ability to rapidly kill cells is really a key to their ability to protect us against the infection. And as I said, that the CD8 T cells and CD4 T cells can persist in humans for decades. And Ramai Pandey, who was part of my lab as a postdoc and then as an assistant professor in the group, really published a wonderful paper in 2017 looking at CD8 T cells after yellow fever vaccination and showed that these cells can be found. Essentially, that paper analyzes how these CD8 T cells differentiate over time, their properties, and showing that key changes in the chromatin structure happen that allow these cells to elaborate function almost immediately. So they're like a loaded gun that is ready to fire because of the different chromatin structure, allowing them to become effectors very, very quickly. And Ramai Pandey now is at Ashoka University as a faculty. We then had a very interesting change in our research. Until now, what I've shown you is the work that we did, and I should also say that others in the field have also contributed to these concepts. One never works in a vacuum. When you're doing something, there are many other groups who are also making important observations. And so all of this was really in the context of acute viral infections. We then turned to the mouse model experiment that we were doing. When we said, well, this is what happens during an acute infection, but what happens if the virus is somehow not cleared and it persists, like you have in HIV and hepatitis B, hepatitis C? What happens then to the CD8 T cell response? And here we made a very important observation. This was work that was done by Alan Zayak, who was a postdoc in the lab. And this cartoon summarizes observations of Alan and others in the field. So this is the course of an acute infection. You get a good effective CD8 T cell response, infection is cleared, and as I showed you, you get very nice memory cells that can persist for decades and elaborate very good effective function. But what Alan discovered was that if you don't get clearance of infection, instead of getting these functional memory cells, you get T cell exhaustion. The important point here was that the T cells are still there, but they are not able to function. So this demonstration of T cell exhaustion opened up a very interesting new area of research, because the fact that the cells were present but not functional immediately allowed us, or the question then immediately became, what is the potential mechanism of this T cell exhaustion? And then the most important applications from that would be that if we can understand the mechanism of this T cell exhaustion, can we develop techniques for restoring function back to those cells? And this led to the discovery of multiple inhibitory receptors in these exhausted CD8 T cells and identification of one particular inhibitory receptor as the major regulator of this T cell exhaustion. We were able to do this because John Veary in my lab compared the gene expression profile of these exhausted cells with these functional memory cells, and this is work that was published in 2007. It actually built upon work that another postdoc in the lab, Sue Tech, had done describing the gene signature of these functional memory cells. So a comparative analysis of the good T cells and the bad T cells allowed us to identify the molecular differences between these two cell types. And one of the most important molecules that was identified in this analysis was this inhibitory receptor called PD-1. PD-1 was originally cloned and defined by Tasuko Hanjo from Japan. And Tasuko Hanjo's pioneering work on the discovery of PD-1 actually was acknowledged by a Nobel Prize 2 years back in 2018. He and Jim Allison got the Nobel Prize for the development of this cancer immunotherapy. But what our work showed was that the major mechanism of this T-cell exhaustion actually was inhibitory receptor PD-1. And this work was published by Dan Barber, who was a graduate student in my lab. And Dan did a very simple experiment, which is using an antibody that recognizes the inhibitory receptor and then asked the question, will this now result in better T-cell function and better control of the viral infection? And amazingly, this very simple monotherapy blocking the PD-1 inhibitory receptor on these cells resulted in increased number of these cells and more function from these cells and reduction of the viral load. After Dan Barber's observations about linking T-cell exhaustion with this PD-1 inhibitory receptor, these findings were very quickly extended by many others in working with human chronic viral infections such as HIV, HCV, hepatitis B, and more importantly, by many people who were working in cancer mouse models and also in cancer human T cells. They also found that that the T cells were there, that is the tumor-specific cells and the HIV-specific cells were present, but they were not functional, and they expressed high levels of PD-1. And these findings actually have resulted in PD-1 therapy in human cancer. This is now licensed for more than 15 cancers and give variable rates, but this is one of the most effective checkpoint therapies. The credit for this, of course, goes to the people who work on cancer and to Tasuku Honjo, who discovered the PD-1 molecule. But our findings with T-cell exhaustion also play a role in the development of this very effective cancer treatment. So I want to end this part by making a few comments about the value of fundamental research. So if this small journey of ours that I summarized to you really started by our lab asking a very basic fundamental question. How do you maintain long-term memory to your infections and to vaccines? A very fundamental basic question. And in that, we made the observations about T-cell memory, about plasma cells, and then when we started saying what happens during chronic versus acute, we made the discovery of T-cell exhaustion and linking it to PD-1. And the application of this PD-1 really ended up being in cancer as a major treatment. And we never worked on cancer in our lab. We never, when we started these studies, when we first asked these questions about memory, we were not thinking of cancer, neither were we thinking of any kind of therapy. We were just asking the fundamental question. So I really want this to be a message to especially the many students that we have and the wonderful researchers that they will come in the future, is try and ask a fundamental basic question. And you just don't know. If you do it well, you don't know where its applications will come. And in our case, the important application came in cancer, even though our lab really never was studying cancer. So let me now one more slide. This is a picture that was taken. It's a dated picture. It's taken about 10 years back or so in celebration of my 60th birthday. And many of these names that I mentioned to you are actually right here. And I've been most fortunate to have really outstanding people join my lab. And the success of almost every lab depends on the people who are in the lab and they do the work. And I've been most fortunate in having truly outstanding students and postdocs who have come to my lab and have now gone on to become outstanding and very successful researchers of the work. So let me now just switch and spend the next 10-15 minutes on COVID-19 vaccines and where we currently stand with this. I don't need to show you this slide, but it's sobering to kind of look at this. That this pandemic that emerged in Wuhan in China in late 2019 has become really the largest pandemic of the century. There are over 65 million cases worldwide, 1.5 million deaths, and it has impacted 190 countries and regions. I think no one in our lifetime has ever seen something like this. Last time this happened was in the 1918 influenza pandemic. And it's really quite remarkable that this virus spread so quickly and has overwhelmed not only public health, but as was pointed out by Honorable Vice Chancellor, really how we live. It has impacted public health as well as economic health in a major way. So the first question one wants to ask is, why did so many people get infected? And the answer for that is very simple. The answer for that is, we're all immunologically naive to this virus. And this is the key. If a pathogenic virus emerges, sometimes the viruses that emerge are not that pathogenic. Unfortunately, in the case of COVID-19, it can cause very, very severe disease, especially in the elderly and those with other comorbidities. And we have never seen this virus. This is how horrible pandemics are. That the virus is highly efficient in infecting, can cause disease, and we are naive to it. We don't have any pre-existing immunity. And this also points out how important it is to get the vaccines. We've forgotten how terrible diseases like measles and many others are. So getting the vaccine really has, as a child, then the child is exposed, which is no longer naive, and that's the basis of vaccine-mediated protective immunity, which I highlighted in the past few slides. So what does this virus do? And what are the current strategies for making vaccines? So this is a picture of this virus, which I'm sure everyone has seen. And this protein, the spike protein, which stands out, really stands out, and looks like a crown. And that's why when all of these viruses in this family, they're called coronaviruses, because when the structure we looked at, it looked like a crown. So this crown is actually decorated by this spike protein. That's what this is. This is the protein that is used to attach to the cellular receptor. And that's highlighted over here. So this is the SARS-CoV-2. I've been using the terms interchangeably. COVID-19 is the disease. It's caused by SARS-CoV-2 coronavirus. And this is the structure of the SARS-CoV-2 spike protein. It has a receptor binding domain. And this is the part that binds to the cellular receptor, which is ACE2. This is actually a very large glycoprotein. Most viruses don't have a glycoprotein that large. That's why it really stands out. That's why the term crown or corona comes up, because it's really sticking out from the surface of the virus. And this region that binds to the receptor is about 220 amino acids. This region is totally unique. There are human coronaviruses, and also, if you remember, there was SARS-CoV-1 and there was MERS from the same family. But this region is very unique to this particular SARS-CoV-2 virus. And that's one of the reasons why we don't have much protective immunity from the human coronavirus, the common human coronavirus. This is a very unique region. And most of the vaccines that are currently being developed are really focusing on producing good antibody against the spike protein. So the antibody would bind here to the spike protein, and in particular, much of the neutralizing antibody is to this RBD region, and that will prevent the infection. And that's why most of the vaccines have the spike protein as the virus antigen there, to which they want to raise antibody responses. Hopefully also some CD8 T cell responses. I'll get to that in a minute. So one observation that was made by us, by the Emory Vaccine Center, Mehul Suthar and Jens Rammert, were faculty members at the Emory Vaccine Center, and also by many others. But this was one of the early findings that was made, and this was a very promising finding. What Mehul and Jens found is that COVID-19 patients, and we had analyzed about 50 patients who were at the Emory University Hospital, some of them are very sick, that very quickly, within 2 weeks in some cases even within a week of a confirmed PCR, but certainly within 2 weeks of a confirmed PCR test, all of them had some levels of neutralizing antibody. This was a very nice result for many reasons. One, it showed that this virus actually induces good neutralizing antibody, that is, it could be controlled. and was a good sign for vaccine development. Saying that this protein, this spike protein, is very immunogenic, and the human immune system can make neutralizing antibody against this virus very quickly. And I contrast that with something like HIV. Very difficult for the infected people to make good nuclear antibody for extended periods. So this was good that one could get very good neutralizing antibody responses quickly. And many others have made similar observations. So now I go to what are the So what are the current vaccine platforms that are being used? I've listed the 5 vaccine platforms that are currently being used. The genetic vaccines, this consists of messenger RNA that can translate and make the S protein after injected, or could be DNA, which will then make messenger RNA and then make the spike protein. So one are the genetic vaccines, where basically the gene that will be either producing it after making message, or the message itself, which will be translated and produce the spike protein. That's one strategy. Another one is having a viral vector that is expressing the spike protein, or you can take the spike protein itself, combine it with some adjuvant, and use that as a vaccine. This, of course, is a very classical vaccine strategy. Many successful vaccines are based on this. These are newer technologies, currently no licensed vaccines that were used in the past. You can inactivate the virus. This again is an old strategy, and also attenuated live vaccines. So how does one actually go about testing these things? Let me go back. So currently, in terms of preclinical studies, there are a very large number. There are probably over 100 preclinical studies going on, testing all these different approaches. But in terms of human clinical trials, this has also gone very fast, and many of them are in human clinical trials. And I'll just give you, show you a slide how human clinical trials are typically done. So the first time one puts a vaccine into human, this is referred to as a phase 1 clinical trial, usually involves 50 to 100 people. The main goal of this phase 1 clinical trial is safety, dosage, and some early look at immunogenicity. One does not get any data on how protective this is, but one gets data on safety, dosage, and immunogenicity. Phase 2 clinical trials, which typically include 200, 300 people in that, sometimes more, sometimes slightly less, but that's the number that's used. And this is an expanded phase 1, but so you again get data on safety and immunogenicity, but also now you get some data on different age groups. So essentially, an expanded phase 1 with more data on immunogenicity, more on safety, and different age groups. And then, of course, the real test of the vaccine is in a phase 3 efficacy trial, which involves thousands of people. So where are we currently with these COVID vaccines? There are over 40 currently worldwide COVID-19 vaccines which are in phase 1. There's about 20 or so which are in phase 2, and there are also many, about 8 to 10, which are in phase 3. And in the next few slides, I'll tell you something about the vaccines which are currently in phase 3, and some of them have reported data and some have not, and I'll just quickly go over it. So there are 4 vaccines which are currently have completed phase 3 or are in the process of completing phase 3 and have reported efficacy data. The first one to report efficacy data was the Pfizer-BioNTech mRNA vaccine for the spike protein. Again, the strategy here is quite straightforward. You inject into the people, the vaccine contains the mRNA that will produce the spike protein after going inside a cell. And the technology here that made this possible is really learning how to deliver this vaccine. And this is put into lipid particles. So the trick here really was the delivery part. And there was a lot of innovation by the people who developed these RNA vaccines and really optimizing it. And this has shown remarkable efficacy. So the early efficacy data is over 90%, actually it's almost 95%, a remarkable result and a very promising result that you're getting over 90 to 95% efficacy with this vaccine. It's given in 2 doses, You get your first shot, and then you get your second shot 2 weeks later. The Moderna vaccine is also a similar platform. Again, it's an mRNA vaccine that will produce the spike protein after it's injected in the body. And again, this has over 90 to 95% efficacy. Same thing with Moderna, there's a first shot, and in this case, with Moderna, 4 weeks later, you get the second shot. Both doses are needed to get the optimal response. AstraZeneca uses a slightly different approach. This is the spike protein inserted into a viral vector. In this case, the viral vector is a chimp adenovirus. The reason it's a chimp adenovirus is because many human adenoviruses already are present in many of us, especially some of the more common ones. But a chimp adenovirus would not have any problem with the vector being clamped down by immunity. So the strategy here is to use an adenovirus which will go in, produce the thing, not be interfered by previous immunity, and produce the S protein. And this one also has reported promising data, and with about 70% efficacy. And the Russian vaccine uses also the viral vector adenovirus approach. In this case, there are 2 shots of the adenovirus given at different intervals, early and then second one a few weeks later. In the case of the Sputnik 5, this is the Russian vaccine. It uses what's called a heterologous prime boost. The first immunization is with Ad26, which again is a rare adenovirus that takes well in humans. And then this is followed by Ad5, which is more common, but actually is a very important booster. So this combination of priming with Ad26, boosting with Ad5 actually is quite good. And this has reported 90%. So this slide is really a remarkable achievement. A remarkable achievement of the pharmaceutical industry, the funders, and the scientists who did this work. Within a year, we have potentially 4 vaccines which have shown very promising efficacy data. In particular, the Moderna and the Pfizer-BioNTech over 95%. The Sputnik, the data there is limited, not that many people have been analyzed yet, but again, it's very promising. And AstraZeneca also up to 60 to 70%. Again, it's a absolutely amazing achievement by these people who have pioneered these vaccines. And this really could be a major turning point in curbing this pandemic. And just yesterday or 2 days back, the Pfizer vaccine was approved for emergency use in the UK, very likely to get approval in the US within this week, within the coming week or so, and also in Europe. So I think that both of these are going to get approved very quickly. Pfizer already has gotten approved, and we'll have to wait and see when approvals for these happen. And in addition to these 4, there are at least 5 others which are in efficacy trials and probably pretty close to being analyzed. One is the Novavax vaccine. Now, this is a more traditional style. Basically, you have the protein with the spike protein, and in this case, they use an adjuvant called matrix and adjuvant. It's combined with this, again, a 2-shot regimen. Efficacy data is not reported, but their immunogenicity data from the phase 1 and phase 2 clinical trials is very promising in terms of good neutralizing antibody. Johnson & Johnson is also very close to completing their efficacy trial, and they use again the viral vector approach with Ad26. There'll be either 1 or 2 shots of Ad26. There are 2 Chinese companies, which are also, actually 3 of them. CanSan Bio is using Ad5 as a viral vector expressing the S protein. And there are 2 Chinese companies, Sinopharma and Sinovac, which are using inactivated virus as the vaccine. So my prediction, and what most people feel, is that within the next 2 or 3 months, you will start getting efficacy data on these. And it's very likely that you may have several different vaccines which are licensed by the middle of next year, or maybe even earlier. So as we move forward, what are the issues? What are the important issues that we have to deal with? So the great success is that we have, within a year, at least 2 2 highly effective vaccines, maybe even maybe 5 highly effective vaccines, and many more. Some of the issues that will come up will be availability and distribution. Availability would mean how many doses are available, how quickly they can be produced, who will get it, how many countries will have it. There are also some issues in terms of the cold chain. The mRNA vaccines, both mRNA vaccines require the cold chain, have been frozen. -20 for the Moderna, -70 for the BioNTech. And this will have some challenges, especially in the developing world. But they seem to be highly effective based on the data. So I think there will be issues of availability, distribution, but the companies are committed to producing hundreds of millions of doses. And so I am an optimist, and I feel that by the middle of next year, if not even sooner, I think that many people will be able to get the vaccines. Then finally, a question that we don't know yet is the durability of this protective immunity. The current efficacy data we have, which is highly promising, is really within the first 2 months after vaccination. How protective will we be a year from now? How protective will we be 5 years from now? Will we need booster shots? So there are many important questions that still need to be addressed. This is my last slide. So we have great fear of pandemics, and we are right in the midst of a horrible pandemic. But unfortunately, some of us also have fear of vaccines, and not everyone takes the vaccines that they should be taking. So I would urge that when these vaccines come out, and depending on what is available where you are, that you should take the vaccines. I will not tell you which one you should take. I don't have any favorite, but I think we should go by what the data is, and so far the data looks good for many of them. And when it's available, we should take it. I wouldn't recommend waiting for the very best one, or the one that might give you long-term protective immunity. You just don't have that data right now. We should take it because the only way we will bring this pandemic under control is by a large number of people getting vaccinated. By that I don't mean 10% or 20% of the population, but 70% or more of the population really should get vaccinated to bring this pandemic under control. And as I mentioned before, and as everyone is aware of, this is not just a public health issue, it's an economic issue for the whole world. So I leave you with these words, that great hope that we have that there will be effective vaccines, and the fact that this vaccines will hopefully turn around this pandemic. Thank you very much for your attention. And happy to take any questions.
Dear sir, thank you so much. It was an illuminating and informative talk on vaccine and immunological immunity. We are also thankful to Dr. Ahmad to share the information on recent vaccines for COVID-19 and their platforms. It is educational to understand the issues as well as challenges in vaccine distribution and the positive optimism that these vaccines share among the scientific world. As Dr. Ahmad asserted that more than 70% of the population should take the vaccine for having the pandemic under control. We are hopeful as well. Please note that all of you can write your queries or questions in chat box, both in WebEx and YouTube, and we will request Professor Ahmed for his comments. Now, I request Professor Zahid Ashraf, Head Department of Biotechnology, who himself is a very active researcher in the area of coagulation and hypoxia, to conduct a question and answer session. Professor Zahid Ashraf, please.
Rafi, can I ask you a question?
Yes, please, Narendra. Please.
Narendra Mehra.
Yes. How are you, Narendra? Please go ahead.
And I like the slide of those 5 decks of 9. And these vaccines are going to be used at all for the first time in humans, right? They've never been used in the past, and we do not know as yet the durability of it is to know more opinion on the use of mRNA-based in COVID-19?
Yeah, then sorry, you were breaking up, but I think you were asking the question about that these are new platforms. Yes, indeed, the RNA vaccine is a new platform, and as I mentioned in my talk, actually is a very creative and innovative way of delivering the RNA that was developed by these people who worked on this. It's a new platform, but so far the data is highly encouraging. And we don't know the question about durability, and that's why I brought that up. Is that something that we will have to wait and see what happens? One of the nice things about the RNA vaccines is that if there is no vector immunity, because there's no vector there, so one could boost as many times as you wanted. So I think
I think effectiveness at the population level.
Sorry, you are well, you're breaking up.
Levels.
Sorry, maybe I can't.
Can I ask you a question?
Yes, please.
It's a very good talk, really very informative. One question with this mRNA vaccines, even if it's general feeling of general masses, but even if after say 6 months, there is no circulating antibodies, even after booster, let us say, but if a virus infects, do you think that it will act as a booster and people can be saved?
Yes, that's a very good question. It's not, so even after antibody levels go down, you still have T cells, and the RNA vaccines should also induce a reasonable CD8 T cell response. So there will be the memory CD8s and memory B cells which could compensate for that.
And for clarity,
Ideally, that will give you long-term antibody responses. The pre-existing antibody is, I would not underestimate it. The faster recall response, the better.
and when you say it's 70% efficacious or 90%, is it that if you give to 100 people, 90 will generate antibody, or is it that 90% of the viral load will be reduced and 70% of the viral load will be reduced?
Yeah, no. So let me give you, I didn't go into how the efficacy was calculated, but I'll give you one example. I'll give you an example from one of the Pfizer studies. Okay? But all the studies are the same. So actually, the way this phase 3 clinical trial is done is really based on disease, not on antibody responses. So in both the Moderna and the Pfizer BioNTech trial, almost 40,000 people were enrolled. Okay. The initial data was based on 40,000 people. Half of them got placebo and half of them got the vaccine. So basically you've got 20,000 people getting placebo, 20,000 people getting the vaccine. And then you see how many infections happen with the placebo group versus the vaccinated group. And the data are remarkable. That when they analyzed this, there were like over 200, almost close to 200 infections were there, 190 or something, and only 8 or 9 were in the vaccinated group. 180 or so were in the placebo group. That's how you calculate the efficacy group. So it's very robust. Same data with Moderna and with Pfizer. The specific data is with fewer number of patients, but again, very promising. So basically, this data is analyzed in very large number of people, and these are all confirmed. So basically, what one does is after giving the placebo or giving the vaccine, these vaccinees are followed longitudinally to see if there are any symptoms. So if they have any symptoms, they go to the clinic and then they're diagnosed whether it's COVID or not. So basically, so of the 40,000 people that were analyzed, 20 in placebo, 20 in the control, in the vaccinated group, there were 90% of the infections, or 95 actually, I'm underestimating the efficacy by 5%, 95% of infections were in the placebo group and only 5 in the vaccinated group. And what's unfortunate is that in the 5% which were in the vaccinated group were not very severe. So there were no severe disease in the vaccinated month. This is remarkable. The data is really something that's given all of us great hope.
Thank you, sir. Thank you.
So I would like to request Professor Shahid Jamil sir to shed some Indian perspective on it. He has been a very active collaborator with Dr. Rafi Ahmed sir, and then has been one of the pioneers immunologists in this country. So I would like to request his comments. Sure, Shahid sir, please.
Rafi, thank you for the wonderful lecture. And it's great to see you. What can I say after such a wonderful lecture? I think most of you will agree that Rafi is a first-rate immunologist, but he doesn't talk like an immunologist. He talks like a normal person, and makes us understand the intricacies of immunology. Yes, we've had a long association, and I spent some time at the vaccine center, wonderful place, and we've been sort of partners in setting up the vaccine center at ICGB. So it's been a wonderful relationship, and which I cherish very much. And even at the cost of embarrassing him, let me just say that he's a very fine immunologist, but he's an even better person. Thank you for being there.
Thank you, Shahid, for the very kind word. Yes, as Shahid mentioned, we've had Shahid has been a friend, a wonderful colleague, played a very big role in us setting up the vaccine center at ICGB. And Dr. Veer Chauhan was the director at that time. So from ICGB administration, Shahid, and of course the PT is very critical in the success of that sector. It continues to be.
We have another eminent immunologist, I would say, the father of immunology in this country, Dr. G.P. Talwar sir. He was in the beginning of, he was there in the lecture. Could you please unmute yourself and giving you the rights for your comments? Dr. G.P. Talwar sir.
Sorry, what was the question?
No, a lot of people are asking the question, the basic, how long the immunity will be there after the first dose of vaccine? That remains one of the multiple questions.
Yeah, could you repeat that? What's the question after the first dose?
Yeah, how long the antibody would be there? That's a general question.
Oh, yeah, right. So then we don't have that data. We don't have much of that data yet. You need the two-dose regimen is needed. After the first shot, you get some antibody responses, but you do need the second shot. If you don't get the second shot, you're not going to get the high level. And how long that antibody after the second shot will persist, we don't know. But there was just a paper that came out, I think, yesterday in New England Journal of Medicine. This is a follow-up for the people who got the Moderna vaccine from the phase 1 clinical trial, and they are reporting data about 4 months after the second shot, and antibody levels are still quite high. So there is not much of a decline, at least in the first 4 months. And the efficacy data is based on anywhere between 2 to 3 months or 2 to 4 months after the second shot. So we know that in this first few months, there is fantastic protective immunity. But what we don't know at this stage, 2 years from now or 5 years from now. But in the case of this pandemic, I wouldn't worry about that. I think we have to take it. We have to take the vaccine and not wait for something that might be, and we don't know. These might also be very long. But that data will we need to wait for that.
Professor Zahid, can I ask a question?
Yeah, sure.
Yeah. A good evening, sir. Thank you so much for the wonderful informative. My question is, certain repeat infections have been reported with COVID-19. So, would you like to comment about these reinfections in terms of memory cells?
Yeah, that's a good question. There have been these anecdotal reports of some people getting reinfected, but I think one would have to wait and see how rigorous those data are. Whether it was essentially not full recovery, but kind of a little bit coming back up. That happens often. You have an infection, you kind of control it, but you haven't really completely eliminated the virus in these first 2 or 3 weeks. So a little bit of, you get that. So I think I would be a little bit more careful about whether how many people are getting reinfected. The current data is that if it's happening, it's very rare. Now, certainly one may get reinfected 2 years or 3 years or 5 years later, because I gave the example of measles where you do get this lifelong immunity, but not every vaccine gives you that, and not every infection gives you that. We know that we can get the flu more than once. So it's not every infection that gives you lifelong protective immunity. So reinfections can happen, but the current reports saying that's happening within the first few weeks or months, I think are more anecdotal than really that robust information.
So, a lot of people are asking about the side effect of the vaccine, and which I think is one should not worry about now. But a lot of people are asking that what could be the possible side effects.
That's a very good question. I didn't mention that in my talk. I was sure that someone would ask that question. So the RNA vaccines do have reactogenicity. So after you get the first shot, you will very likely have some pain in the arm. You might have a rash. You might have some fever, some headache. And interestingly, after the second shot, the reactogenicity is even greater. So it's very likely that almost all of us if any of us gets these RNA vaccines, we will be in some pain for a couple of days or a few hours after the first one, and maybe for a little bit longer, maybe 2 days, 1 day or 2 days after the second shot. So these are fairly reactogenic. They're more reactogenic than most of the vaccines that we typically take. But so far there's no data of anything really serious in terms of I'm making a distinction between reactogenicity versus really serious adverse events. Reactogenicity is something that happens for the first couple of days, or a day or 2 days, 3 days maximum. But typically for 24 to 48 hours, and then you're okay. So the mRNA vaccines do have that reactogenicity. But so far, no serious adverse events have been reported. With something like the protein plus adjuvant vaccine, the reactogenicity is less than the RNA vaccines. The ad vaccines, reactogenicity is not that high, but there have been a few reports of, with the Oxford vaccine, there were 2 cases reported in Europe of transverse myelitis, which is a neurological disease. So, a small number, but something we will have to be watching out for. But that would be more concerning than the reactogenicity. Reactogenicity is some discomfort that we will have for 1 or 2 days after getting the vaccine.
Sir, other couple of questions, I'm just summarizing a lot of people's questions.
Sure.
Is that the other thing they are asking that about the COVID, what are the other organs which are being, so far a lot of people, they know the lung is the major organ which is infected.
Yes. Yeah, that's a very good question. So there are some interesting things so the primary infection, let me take a step back. The primary infection is of the lung. The virus gets in through the respiratory tract. If the infection is of the upper respiratory tract, then the symptoms are much milder. as you know, there's quite a bit of gradation in the disease symptoms. Some just get very minor illness, just flu or cold-like symptoms. Those individuals, infection is really just on the upper respiratory tract. But when you get the more severe disease, it goes deeper into the lung. So once it goes into the lung, that's when the problems happen. Lung remains the major target organ, but this has some peculiarities that people don't understand. There are these clotting issues that happen. The precise mechanism for that is not understood. But there isn't any compelling data that many other organs are infected, although there's a lot of discussion about it. Studies that have been done on autopsy material. Some of this was done by Sherif Zaki, an outstanding pathologist at CDC, and he showed that the major organ that's infected is the lung and the respiratory tract. So he did not find a large amount of virus in the other tissue. There's some evidence that might be a little bit different, but again, it's mainly a respiratory infection.
I think
But there are some unusual symptoms which need to be, which need to be studied.
So there is a question from the other set of multiple questions from the big. Will it be really advisable to take multiple vaccines to increase immunity?
I wouldn't do that because I think we should take, I wouldn't initially try and make your own combination vaccine at this stage. I think we don't know. I would take the vaccine that's available and has good record of efficacy and safety. I think these are all done by reliable authorities, do this analysis. So what's available to you in India and has good efficacy and good safety record. I would not worry too much about the reactogenicity part, but I would make sure that it's safe in terms of other more serious adverse events and has shown efficacy data. I would just take that vaccine and not try to take multiple vaccines.
There are several other questions, but I'll forward you, and then we have noted the email ID, and then we can forward them.
Yeah, please. Please feel free to contact me by email. I think many of people on the call have my support. Please feel free to share my email with anyone who wants to ask a question. I'll be happy to answer those questions. Yes, Vice Chancellor, you're raising your hand.
Yes, sir.
Can I ask one question? MM Khan?
Vice Chancellor is saying something and I can't hear. She might hear. I think she's muted.
Yeah, I am MM Khan. I am asking that is there any study about the rural and urban infections? That means the pupil affected in rural areas as well as the urban areas. Is there any study like that?
No, I mean I don't know that. It would be interesting. I mean, I'm not that familiar with the details of the vaccine trials being done in India, COVID-19 trials being done in India, whether they're including rural as well as urban. That's an important question because I think we do want some data within a large clinical trial on rural and urban areas.
Thank you.
Yeah. Thank you, sir. I think most of it has been that is.
Sir, I would like to have the last and final question from the Vice Chancellor, ma'am, and to conclude the question answer session.
No, I was just trying to say that it has been a very informative and a very interesting lecture, which made me sit through the whole lecture. I came only at the beginning, but it was so nice, and it was definitely a very nice person that you, Professor Zahid, you have introduced to us. Somebody whom we do not feel that you are speaking from some other place, and you are such a very well-known researcher. You feel that you are one of us. So I again invite you to be part of Jamia in any way, a loose connection or a firm one, but I am also interested to know what your center can do for Jamia and what Jamia can, how we can reach out to you.
Thank you. On my next, I come to India at least 3 times a year, but of course this year I have not traveled. But on my next visit, I will definitely come and visit Jamia and hopefully get an opportunity to meet with you and the other people.
Thank you very much. Indeed a pleasure.
Thank you, wonderful, sir. We definitely want to keep hearing you, but I think in the interest of time, I'm wrapping up the interaction session, and we will, as ma'am has mentioned, we continue our. This is the beginning of our association, I would rather say. And then it's so kind and nice of you, words are not enough to thank, appreciate your timing and getting early in the morning to catch up with us, and then being so humble and polite. With so many generous blogging days and technical glitches and all those. So I will now hand over back to Dr. Asta Mishra to continue the proceedings over from our question. Thank you, Avis.
Thank you, VC ma'am. All the questions and making us understand about the effectiveness of vaccines and their side effects. We are not obliged to have multiple companies of all equipment. And now, I would like to invite Professor Vasir ma'am, the Faculty of Natural Sciences, for the vote of thanks at the conclusion of our event. Professor Siri is an esteemed immunologist who has authored 2 books and several research articles. I would now like to invite him for the vote of thanks. Professor Siri, please.
Thank you, Dr. Asta. It has been a pleasure, Dr. Rafi Ahmed, to listen to you. And I see a lot of renowned immunologists from India here on the panel today, waiting and hanging on to your words, and who have been giving us a ray of hope, and being the optimist that you are. I find very much in league with you because I'm also an optimistic person. And we have, and the only thing that I fear is not the reactivity, but the cross-reactivity, not the reaction that a side effect that this vaccine would do, but we just hope that we do not have any cross-reactivity that can give us far, intense situations. Of course, we need to have some more time to see the efficacies and so on. But right now, my pleasant duty is to thank you, Dr. Rafi. And this is on behalf of everyone who has been involved in organizing this Centenary Lecture. I would like to thank you, Dr. Rafi, who's the director of Emory Vaccine Center, Atlanta at US, who ignored the huge time differences and delivered to us a very informative talk on the topic immune memory, remembering your infections and the vaccines. Dr. Ahmed's talk and his scientific expertise is truly inspirational and enlightening too. It is also very exciting to see the developments at the Emory Vaccine Center, from which the entire mankind will be greatly benefited. I also feel very overwhelmed to see that there are over 1,000 registrations from all over India, and an equal number of them logged in to listen to this lecture. And to the participants, I hope that you all found the lecture very informative and that you spent your time listening to it. You will find it very, very useful for your information, for your life, and everything for your future. Now, this has been a very enriching experience for us at the Department of Biotechnology at the Jamia Millia Islamia, while organizing this lecture as a part of the Centenary celebrations. We have had many positives to take forward from this lecture. I thank you, Dr. Ahmad, once again. We extend our gratitude to Professor Najma Akhtar, our Vice Chancellor, under whose vision and leadership we have been able to manage our centenary celebrations successfully during these very difficult times. All the administrative staff of Jamia Millia Islamia, including the Office of the Registrar, and the finance officer. They extended exceptional support in making this event successful. We are all truly thankful to them too. And how can I forget to thank Professor Zahid Ashraf, the head of the Department of Biotechnology, who has also been my student for his tireless efforts and contributions in organizing this lecture, along with Professor Mahfuzul Haq, another student. Jamia also acknowledges the entire faculty of the Department of Biotechnology for their continuous support and encouragement in bringing together everything for the successful conductance of this lecture. A special thanks goes to Dr. S. Kazim Naqvi Sahab. He is the officiating director of the Center for Information and Technology of our university, and his team also for the technical support that was required for this webinar. And this was supported by Dr. Asimul Islam for assuring a glitch-free event. Last but not the least, we thank the phenomenal support of all the office staff of the Department of Biotechnology, our students and the PhD scholars whose extraordinary momentum and incredible zeal which made it possible to accomplish this event. I wish everyone safety and health during these unprecedented times. Thank you very much, everybody.
Thank you. Thank you for those very kind words. This truly was a privilege and an honor to be part of your centenary celebration.
You are most welcome, sir.
Please show the picture again, the portrait. It is, and also thanks to the Fine Arts Department for providing this picture.
That looks very nice.
So thank you again.
Thank you, sir. Thank you so much. Now you have to come to Jamia to collect this. Thank you so much.
Thank you so much.
Thank you. Thank you to all of you. Really, you were most wonderful.
The media interaction. Sorry to bother you, keep continuing bothering you.
No.
Media interaction, that is on the different platforms we will meet. Thank you very much, sir. Once again, thank you very much, DC ma'am, for staying till the end. Thanks again for Professor Shahid Jamil, Professor Chibi Talwar, Professor Nehra, all those iconic immunologists, Professor Kazi, there are a lot many immunologists, and I think they are holding, they are continuing, are supporting the efforts to develop, not only encourage the researchers to take immunology as a mainstream, to translate their world vision or translate their societal need for developing vaccines and pursuing immunology, not only on the translational front, but also on the fundamental. Thank you once again all. Thank you. And we leave you with the memory of Professor Ahmed's wonderful portrait made by Fine Arts faculty. I think.
Thank you.
Thank you.
Bye-bye. Thank you.
Bye.